Starting off, congratulations to Onyedika for passing his POTS. Actually, class on that. Brian also got his dissertation approved. So I think avoiding attention. And what else do we have? TV Day. So next week, many of the students will be presenting on cardiovascular. Good luck to them as well. That's it.
Okay. So I get the honor of introducing our speaker today. I hope a lot of you got the opportunity to meet with Dr. Smuder. Really incredible career trajectory. She's currently an assistant professor, but you wouldn't believe it if you looked at her CV. The other thing I think many of you may have noticed that was really interesting is she started at the University of Florida, and she's really come full circle, and she's at University of Florida as a faculty member. She did her bachelor's degree in exercise physiology, or received her PhD in exercise physiology at the University of Florida. She then transitioned labs for her postdoctoral training, did a short bout of a faculty position at University of South Carolina. For those of you that understand how you submit R01s, you get something called early stage investigator status. So the first time you submit an R01, you get a little bump in your score. She submitted 2 at the same time. They both got funded. So yes, it's allowed.
Can you be early stage twice?
If it goes in the same cycle, you can. So if you have high energy and good ideas and you're an early assistant professor, you submit 2 R01s at the same time, both good ideas, they both get funded. And then she took her lab and her great students and she went back to Florida and got another R01. So I think what that speaks to, and she studies exercise science, which I just think is wonderful because a lot of us think NIH doesn't fund exercise. But Dr. Smuder can tell you they do fund exercise a lot. And if you have good ideas and you apply them to a really relevant population. And so she's looking at not only respiratory function, but cardiac function in chemotherapy models in cancer. And so just get an amazingly successful person, really fun person to talk to. Really interesting research area that we don't have a lot of on campus. And so I'm really happy that she's here and I won't talk anymore. Oh, she's also top 40 under 40 at the University of Florida, other than other awards, but that was one that was really cool too. So thank you so much. We appreciate you being here.
Oh, great. Thank you. I really appreciate the invitation. It's been a great day talking to so many of the students and faculty members, and I'm really happy to share some of this work looking at exercise and chemotherapy and what my lab is currently doing. And so with that, today we'll kind of hit 4 major topics. I'm going to introduce you to what chemotherapy toxicity is and the chemotherapy that my lab focuses on. I'll show you some of the clinical evidence that exercise is good. And then we'll look at some of the basic science that my lab is actually focusing on in discovering what are some of the mechanisms of exercise protection. And then some of the new work that we're doing in trying to better model chemotherapy and exercise in our animal models so we can hopefully help to translate some of these exciting findings that we have. And so what this graph here is really showing is a very positive effect that we've had over the last kind of 50 years. And all these arrows pointing to the right show that through advances in screening and early detection, as well as beneficial therapies, we've actually had a large increase in 5-year survival in cancer patients. And this is very exciting. It's also causing concern for the patients that have then been treated with different types of toxic chemotherapies, radiotherapy, and different things like that. And so they're actually then having a worsened quality of life based on a lot of the toxic effects of these chemotherapies. And so in general, while there have been great advances made and while we do have the increased survival in these patients, what most patients end up getting is a cocktail of chemotherapy treatments. And so we have some of the examples of the classes of chemotherapy treatments here and the way that they're able to target different areas of cell replication in order to inhibit that and cause cell death and reduce cancer growth. However, what we end up seeing though is that each of these types of classes of chemotherapy agents also come with some very negative off-target effects. And so they vary depending on the target of the chemotherapy. We see a lot of neural toxicity, a lot of cardiovascular toxicity, myelosuppression, increased allergic reactions. And so what my lab focuses in then is this specific class here, the anthracyclines. And so this is a highly effective class of chemotherapeutic drugs. They're used in a lot of gynecological and blood cancers. However, even though they're highly effective in what they do, they cause a lot of very negative adverse effects. So in general, it's estimated that these negative effects or the positive effects to cancer outweigh the negative effects. And what we see is that there's similar mechanisms in the way that they cause apoptosis of the cancer cell, is they're also able to directly target the cardiac tissue and the skeletal muscle tissue to also cause severe effects. In general, we have both acute and chronic effects. For cardiotoxicity, more acutely, you end up seeing a lot of different arrhythmias and hypotension. And more chronically, we end up having these patients develop congestive heart failure and left ventricular failure. For myotoxicity, this more is looked at as far as fatigue in these patients, exercise intolerance, and in general, just a reduced ability to perform their activities of daily living. And so it's actually been well known for a long time, and really since doxorubicin was discovered, that it can cause cardiotoxicity. And this same experiment has been shown several times where what both in 1979 and 2003 we see is that there's a very large dose response as well as an age response to your development of cardiovascular dysfunction or having some kind of cardiotoxic event in these cancer patients. So you can see here that we kind of have this influx that we get around 600 milligrams per meter squared of a cumulative doxorubicin dose, as well as the older patients are the ones that are more highly affected and have more adverse effects from doxorubicin treatment. And because of this, really the only intervention right now to reduce cardiotoxicity is to reduce the dose that patients are receiving. So they really try and cap the dose at about 500 milligrams per meter squared total dose for patients. If we look at skeletal muscle, we also see similar adverse effects as far as the fact that these are cross sections of a rodent diaphragm muscle. And you see that you have a lot of infiltration of connective tissue, you have a loss of muscle fibers, and you have atrophy of the fibers as well. You can also see that you have mitochondrial dysfunction and again, just a worse ultrastructure of your skeletal muscle from doxorubicin. And it's really well established that doxorubicin itself can just cause muscle cell death. And it's been used for different types of facial spasms where they'll directly inject the doxorubicin into the muscle in order to basically cause apoptosis and cell death. And we can see this here in these examples where we have our two control muscles and the ones that have been treated with doxorubicin. You have a reduction in cell fiber size as well as number, and this increase in connective tissue and just a loss of muscle mass in general. And so we're able to look at a lot of those things in animal models. Unfortunately, the primary way that any of the muscle dysfunction has been shown in patients is through a lot of these different types of surveys. And so they'll really just try to figure out how much the patients are fatigued, how well they're able to perform activities of daily living, and kind of how much they feel like these deficits are affecting their life. And so what we can see here, though, is that we do have a very strong shift of an increase in fatigue score in patients that have been treated with chemotherapy. Some studies are really trying to find other ways in order to assess muscle dysfunction in patients, and this is just a couple examples. The top one shows a hand grip strength test, and they have a reduction in grip strength in the patients that were treated with the doxorubicin-containing chemotherapy. There was actually one patient study done where they had an isolated limb perfusion with doxorubicin, and they were able to take biopsies prior to and following the patient treatment, and they did see atrophy of the limb muscle fibers. And then finally, the most common way that kind of muscle dysfunction is looked at in patients is through a lot of 12-minute walk tests. And so this shows for breast cancer patients kind of what their baseline distance was for 12 minutes. And then this is actually 6 months following the completion of therapy. So they've had time to kind of rehabilitate from therapy. And you still see about a 10% decline in these patients that have received a doxorubicin-based chemotherapy. And so a lot of work has really been done to try and figure out what are the mechanisms of the toxicity within both the cardiac and skeletal muscle. And they do seem to be very similar in the way that we see the dysfunction occurring. And generally, there's some of the primary determinants that our lab has looked at, and that in general, the community agrees are ways that we have this muscle dysfunction occurring. Doxorubicin is nonspecific to tumor cells, and so when it is infused, it does actually accumulate in tissues with a high mitochondrial content, because it does have an affinity for cardiolipin, and it ends up causing redox cycling, increased reactive oxygen species production, and mitochondrial dysfunction in both cardiac and skeletal muscle. We also see that we have activation of proteolytic activity of autophagy, ubiquitin proteasome pathway, as well as of calpain and caspase-3 that all are contributing to muscle breakdown. We do see that there are actually sex differences between patients. It varies a little bit depending on age, where younger females as well as postmenopausal females seem to be more affected, whereas middle-aged males actually seem to be more affected. So kind of changes based on the way that sex hormones change over the lifetime in both males and females. And then finally, one factor that is particularly of interest of our group is really looking at the metabolism and clearance of doxorubicin. Doxorubicin is given based on the total mass of a person, so their meters squared, their volume. And with that, it doesn't matter the body composition of the patient, it's really just given based on that number. However, doxorubicin itself does not accumulate in fat mass. And so if you have two people of identical body mass, the one that has more lean mass would actually have less toxicity compared to the one with less lean mass, because you're gonna have more accumulation in that lean mass compared to the fat mass. So it makes a big difference. And there's a lot of people who really think that the body mass index of the patient really needs to be taken into consideration for dosage rather than just the total body surface area. And so with that, there really are no approved therapies to treat doxorubicin cardiotoxicity. A lot of work is starting to be done looking at exercise, and this is because, as you all know, we see a lot of really positive benefits of exercise that go beyond even just looking at cardiovascular health and the skeletal muscle. In general, in cancer patients, you do also see a reduction in the weight gain that they tend to have. You have a lot of cognitive dysfunction that can occur with chemotherapy as well. You have different types of nausea, fatigue, and other kinds of stresses in patients. And so really exercise seems to be this, full body positive therapy that can not just protect your cardiac and skeletal muscle, but may be of overall benefit to the patients in general. More and more work is being done looking at exercise in patients. However, there are a lot of limitations as far as the ability to prescribe the right exercise, to get the right population. With that, a lot of work continues to be done in both males and females, looking mostly and unfortunately primarily just in breast cancer patients. It's another factor that kind of limits the ability for this field to move forward quickly, is the fact that there are so many different types of cancers, types of chemotherapy treatments, and a lot of different factors that really can limit the progression of this field. But what we do see if we look at kind of an analysis of the papers that are out there, so if you look at VO2 peak in females that either receive usual care or exercise trained, that in general, we do see positive benefits of exercise in these patient populations. And so this is data from a review that we put together a few years ago. And so you see VO2 peak tends to be reduced with usual care, and that exercise training can maintain and in some cases increase VO2 peak, as well as increase or maintain their left ventricle ejection fraction. Looking at a few of the studies that kind of try and focus more on skeletal muscle, you see more of these positive benefits. Again, but skeletal muscle is really less focused on within this field compared to the cardiac tissue. But just to kind of summarize a couple of things that were found is that in a combination therapy where patients exercise for 8 weeks, 5 sessions per week, that they were able to reduce their fatigue and improve activities of daily living. In the second study, they had improved cardiopulmonary function. And if you remember back to where I said our usual care patients had a reduction of 10% in their 12-minute walk test, this study also looked at the comparison of aerobic and resistance training, and they showed that these patients who had an exercise prescription during their treatment were able to, with aerobic exercise, have a 25% increase in distance, and with resistance exercise, about a 4% increase. So definitely benefits to these patients' ability to perform activity. And so while this is all really great and positive effects, one of the things that also limits this field is the fact that there are so many patients that have different comorbidities, that have accelerated age, and that have reasons why they cannot or will not exercise. And so one of the focuses of my group has really been to look at exercise and the mechanisms involved in exercise protection in order to see if there's maybe something that can then be targeted instead of patients necessarily having to exercise. And so this work all started a while back, and we really just took some of the previous literature looking at exercise preconditioning with VIDD and ischemia reperfusion injury, and tried to model those in an acute model of doxorubicin cardiorespiratory muscle toxicity. So what we did is we chose a 2-week preconditioning period. So these animals were, and these are all female rats for all these studies. They went under a 5-day habituation period. Then they went 2 weeks, 5 days each week of a preconditioning protocol, about 70% VO2 max for 30 meters per minute for an hour a day. They were given a day of rest, and then they received their doxorubicin treatment, which was just a bolus, one-time IP injection of doxorubicin. And then we harvested their tissue 2 days later. And the first thing, excitingly, that we saw was that this 2-week preconditioning period was able to significantly attenuate the cardiac dysfunction that we see with this acute bolus model. So we can see down here that our doxorubicin causes a reduction in our fractional shortening, as well as an increase in our myocardial performance index, our primary markers of cardiac dysfunction that we get through our echocardiography. And that when we exercise train these animals, we had a complete attenuation of these negative effects of doxorubicin. When we looked at skeletal muscle, we looked at our cross-sectional area of our diaphragm, and we were able to then attenuate, in this case with our exercise, the reduction in cross-sectional area in all 3 fiber types of the diaphragm, as well as we saw an attenuation of the diaphragm dysfunction when we looked at force production along our force frequency curve. Going back to what we consider some of the mechanisms of doxorubicin toxicity, we took the mitochondrial fraction from these animals, and we looked at their ROS emission as well as their RCR. And so within some of those mechanisms of toxicity, we were able to reduce those negative effects by reducing in both the heart and the diaphragm, ROS production, as well as mitochondrial dysfunction. So we have this great model where it led us to say, all right, what are these mechanisms? How are we protecting the diaphragm? How are we protecting the heart and the mitochondria? And so we started to look at a lot of the kind of basic exercise factors that you think are protective mechanisms. And we've done a lot of different studies looking at kind of gain and loss of function in these things to really try and hone in on what is happening here. And so through this, I'll kind of summarize some of the things that we've seen to focus more importantly on what we think is going on. And for antioxidant capacity, we really basically eliminated this for the fact that we have seen in our model and in others exercise protection where antioxidants were not increased, meaning they were not required. With the heat shock proteins, when we exercise trained animals in the cold, we did not see an increase in HSP72, but we also did not protect our mitochondrial function. So we really think that HSP72 is not required. With nitric oxide and mitochondrial turnover, we saw similar things as antioxidant capacity, where you were able to protect these animals without necessarily upregulating these protective pathways, which leads us to our last box here, our membrane transport proteins. And so this is a field that we're pretty excited about because doxorubicin is, transported into the cardiac and skeletal muscle tissue. And based on a couple of projects that were occurring before when we got interested in this, we think that potentially we are reducing the amount of doxorubicin that is being accumulated within these tissues. The first idea of this came in 2013 by a group where they showed that when they either did preconditioning treadmill running or wheel running, that they saw a reduction in the amount of doxorubicin within the left ventricle of these animals. A couple years later, they performed this same study. However, this time they used tumor-bearing animals. And so what we see in the graph on the right is that they were able to show again that there was a reduction in doxorubicin accumulation in the left ventricle. And excitingly, they showed that this does not occur within the tumor. So you weren't reducing any potential anti-tumor effects of exercise or of doxorubicin with the exercise training these animals. In fact, you kind of had this trend for a potential increase in doxorubicin that's localizing to where you would like it to localize to. And so this got us really excited. And so we looked in our heart and our diaphragm to say, can we see this in our model? Do we see this in our model of a 2-week preconditioning? And so the simple answer is yes, that when we look at our heart and our diaphragm and the doxorubicin concentration, we also saw that our 2-week preconditioning caused a reduction in doxorubicin content. And so the question then is, how is doxorubicin either getting out or how is less coming into these tissues? And so the family of proteins that we are looking into right now is this ATP binding cassette transporter family. And so this is a pretty large family of proteins. However, it is well established that there are 8 of them that doxorubicin is a known substrate for. This is where we started this work to say, it must be one of these that are really helping or being affected by exercise in order to change doxorubicin localization. And so we just did a simple test where we took our model and we looked at the effects of exercise, and a lot of genes on here, but the take home is that these do not appear to be affected by exercise. They do not appear to be playing a role in why we have reduced doxorubicin accumulation within our diaphragm and heart. So the question was then, all right, if it's not these 8 proteins, what could it possibly be? And before that, real quick, this was also shown with a couple of them by that by another group as well, that they also, looking at just 3 of them, did not see a specific exercise effect. So again, we're really kind of taking those guys out of the picture. And then the next question, though, was we're focusing back on the mitochondria. And we know mitochondria are very important for doxorubicin. We know they're very important for diaphragm and heart health. And we did a simple experiment where we ended up taking the cytosolic fraction versus the mitochondrial fraction and measuring the doxorubicin content. And really interestingly, what we saw in both the diaphragm and the heart was that if we look at the cytosol, we don't have any difference in the concentration of doxorubicin in our exercise versus our sedentary animals. However, when you look at the mitochondria, as we know, the mitochondria accumulate more doxorubicin than the cytosolic fraction does. And this is precisely where we were actually seeing the reduction in doxorubicin. So then the question was, are there ABC transporters in the mitochondria? And then the answer is yes, and there are actually 4 of them. These are the 4 mitochondrial localized ABC transporters, ABCB6, 7, 8, and 10. They're not well described, and so it's not well known whether they are actually transporters of doxorubicin. And so that is something that we are focusing on trying to figure out. More importantly, though, what we did see when we looked at the gene expression of each of these is that we do actually have an effect of exercise in our exercise doxorubicin animals. So we do have an exercise-induced increase that is maintained with dox. And so potentially they could be playing an important role in exercise-induced protection against doxorubicin toxicity. And so this is really one of the major focuses of the lab right now, is to figure out what these 4 proteins are doing and what their responsibility is. And so right now, we've just started some work looking at right now only ABCB7, 8, and 10. And this is some work by 2 excellent graduate students in the lab, Imtiaz Dalla and Vivian Dorr. And what they did is they took our same exercise preconditioning model, and each day during exercise, these animals were either treated with an antisense oligonucleotide directed towards ABCB7, 8, or 10, or they received saline as the placebo control. And so what we see here is that when we look at just the weight change in these animals, we have weight loss in the animals that receive dox. We have an attenuation of that with our exercise protection, and we lose that protection with both ABCB7, 8, and 10 knockdown. If we look at their heart to tibia length ratio, we see a similar pattern, that we have a reduction in this ratio with our dox-treated animals. It's protected with exercise, and we lose that protection with our treatment of or knockdown of either ABCB7, 8, or 10 with exercise. The only thing we didn't see the same positive benefit with was our runtime to fatigue. And so in this case, we actually had a reduction with our dox. However, we did not lose that protection with either the 7, 8, or 10. And so looking at diaphragm function, this is kind of where these projects are right now. A lot of data is being collected to figure out what happens to cardiac function and all kinds of other fun things. But interestingly, when we look at our force response of our diaphragm, starting in our upper left here, this is the ABCB7 knockdown with exercise. And so we still have our normal function with our exercise and sedentary controls. We have our attenuation of dysfunction with our exercise doxorubicin treated animals. We have dysfunction with our dox treated animals. And when we give that ASO towards ABCB7, we lose all protection of our diaphragm. If we look at ABCB8, we see a very similar trend that we lose all protection of the diaphragm muscle when we knock down ABCB8. However, when we look at ABCB10, we actually see the lowest response to its knockdown. So we still have some loss of protection when we knock down ABCB10, but it does not appear to be playing as strong of a role in exercise protection as the other two. So as I said, there's a lot more work that needs to be done here, but this is all pretty new, and we're really excited about seeing the difference with these proteins. From here, though, we're also doing this, our loss of function with exercise. We are looking at each of these proteins individually to see if we overexpress them, can they protect? What do they do on their own? Are they sufficient for protection? And so the one I'm going to talk about today is actually going to be ABCB10. So it seemed less important for exercise, but it's kind of the one that we've focused on most currently. And so ABCB10, going into a little bit more detail about what it does, what it's hypothesized to do, is as we hypothesize, we believe that it can potentially be an exporter of doxorubicin from the mitochondria. It's believed to potentially play a role in ROS detoxification through a glutathione mechanism. It actually forms a complex with ferrochelatase and ABCB7 to help transport heme out of the mitochondria, as well as through a complex with ferrochelatase and midopherin 1. It's believed to also help to import iron into the mitochondria. So potentially through these mechanisms, overexpression of this protein could provide protection, and it could be part of its effect with exercise as well. So the study that we did here, again, was just a very simple acute study where these animals received an AAV that was had an MHCK7 promoter to specifically target the cardiac and skeletal muscle. They received that as a tail vein injection. They sat for 4 weeks. They received a bolus of doxorubicin. And so what we saw here was that we had no changes in weight between groups over this time. If we look at their body weight change, we did actually have a reduction with both of our dox groups, so we didn't see any protection there. We also didn't really see any protection with our heart to tibia length ratio, and we saw no difference in our soleus or EDL to body weight ratios. However, when we looked at our force production, we did actually see some positive effects. We saw that we were able to attenuate much of the dysfunction of the diaphragm when we overexpressed ABCB10 in the diaphragm, and that we were able to reduce some of the diaphragm fatigue that we see running a fatiguing protocol also on the diaphragm. Looking at cardiac function, again, with doxorubicin, we see a reduction in our fractional shortening percentage, reduction in our posterior wall shortening velocity, increase in myocardial performance index. And in this case, we see a really positive effect of the ABCB10 overexpression to protect against cardiac dysfunction in all of these 3 markers. And so then looking at these mechanisms that we propose are why ABCB10 overexpression could protect, the first thing that we focused on was the mitochondria in general. When we overexpress this, do we have healthier mitochondria? So we isolated mitochondria from the heart and diaphragm of these animals. Again, we see our dysfunction of our RCR with our doxorubicin treatment, and we were able to improve mitochondrial function in these animals. So then our mechanisms, first looking at our doxorubicin concentration. We isolated the mitochondria here and did HPLC to look at doxorubicin concentration in them, actually in both the cytosol and the mitochondrial fractions. And unfortunately, what we see here is there is no difference between these groups. We have a lot of variability, but in general, we don't see that trend of a reduction in doxorubicin in the mitochondria or the cytosolic fraction of these animals. So potentially ABCB10 is not an exporter of doxorubicin. And so then the question is potentially which of these other mechanisms are why we're seeing this protection of the cardiac and diaphragm muscle. And so then focusing on our iron-associated mechanisms, this is really where it appears that ABCB10 could be playing a larger role. If we look at our ferrous and ferric and total iron concentrations. In the diaphragm, we don't see an accumulation of iron in the mitochondria with doxorubicin, but we do actually see a reduction in our Fe2 and our total iron then with our ABCB10 expression. So potentially we're reducing ROS production by reducing our iron within the mitochondria, because doxorubicin can form a complex with iron that is highly reactive and can cause mitochondrial dysfunction and ROS production. In the heart, though, we do actually see an increase in our Fe2 with our doxorubicin, and this was attenuated in our animals that received ABCB10. So we do have potentially a similar but slightly different mechanism between these two muscles. Oops. And then finally, looking at some of these other markers that we have these complexes of ABCB10 with, we have our increased expression of ABCB10. And then we were curious if whether we were seeing a kind of a reciprocal change in ABCB7 because of its known association with this protein. However, we were not seeing this, so it doesn't appear to be an ABCB7 based effect. We do see an increase in the diaphragm of our importer of mitoferrin that wasn't affected by the overexpression, and we have no changes in the heart. And then with our ferrochelatase, that's also part of this complex, we have a reduction with doxorubicin, and we did not attenuate that. So we're not sure what the mechanism here is yet, and we have some other key proteins that we're excited to look at. But in general, it does appear that ABCB10 overexpression in this acute model, and actually a more chronic model that we've done, we're able to protect. And so really, we're just trying to do a little more work here to figure out why. The last portion of the ABCB10 work, though, that we've done is we also, independent of exercise, wanted to see if ABCB10 knockdown was going to exacerbate doxorubicin cardiac and skeletal muscle dysfunction. So in this case, these animals received that same antisense oligonucleotide, but no exercise. They received it 4 days in a row prior to euthanasia, with their doxorubicin occurring on day 3. And so what we see here then is that we had no real effect on body weight change in either of our, when we knocked down ABCB10 with doxorubicin. We didn't have any exacerbation of our heart to body weight ratio. And again, no differences in our soleus and our EDL to body weight ratios. If we look at our force curve, we do lose any protection that we saw with our overexpression when we knock it down. However, we did not exacerbate it when we knocked down our ABCB10. So again, kind of similar to what we're actually seeing with our exercise is when you increase it, it seems good, but when you reduce it, it doesn't seem to have as large of an effect as some of the other ABC proteins. Again, same thing with our fatigue here. We have no protection, but we don't exacerbate it greater than our sedentary, or not sedentary, our control dox-treated animals. Same kind of results with our cardiac function. We have our dysfunction with our dox, and we do not exacerbate that either when we reduce our ABCB10 expression. Looking at our mitochondrial function, again, similar patterns here, that we do not have any significant effect of ABCB10. And then this was what we were also curious about. Would we have increased accumulation of doxorubicin in the mitochondria and cytosolic fraction of these animals when we knocked down ABCB10? We were really hoping that we were going to see this kind of nice gain and loss of function experiment where one had less doxorubicin in the mitochondria and the other one increased it to kind of show its role as a dox transporter. But again, we're not seeing that in these animals. So this really kind of helps to confirm that ABCB10 does not appear to be a transporter of doxorubicin. So it doesn't look like it's a substrate for it. And then finally, looking at some of the proteins involved, again, we didn't really see any exacerbation of these effects. I will point out, though, we really only had about a 50% reduction in ABCB10. However, there's been some other studies that showed that this is really sufficient to actually cause dysfunction in other conditions. So we do think that that is not a factor of why we're not seeing that exacerbation with our model. So real quick then, I'll kind of summarize everything that we talked about in this part 1. I know it's been a lot, but really hopefully you've come home with the fact that doxorubicin is highly effective and that it is limited due to off-target toxicity. So that convince you that exercise is an effective therapy to reduce both cardiac and skeletal muscle toxicity in doxorubicin treated animals and patients. And that we still believe that exercise-induced protection may occur through increased expression of the mitochondrial transport proteins. We just still have a lot of work to do to really figure out, are they actually transporters of doxorubicin, or potentially are they protecting through a more iron-related or ROS detoxification role. And then finally, overexpression of ABCB10, while it didn't have as great of an effect with exercise, it in itself may be an effective intervention to prevent doxorubicin-induced cardiorespiratory toxicity. And so what I'm going to transition to now is really kind of the work that our group has been trying to pioneer over the last couple of years, and that it's taking the model that we've been using with doxorubicin, and it's been taking that bolus dose and really trying to modify it to make it into a more translational model. And so what we know is that, again, dox causes cardiorespiratory dysfunction. It can reduce tumor burden, and that it also causes kind of a lot of these changes to body comp, skeletal muscle weakness, and fatigue. We know that exercise training is able to increase cardiorespiratory capacity, quality of life, reduce fatigue, and risk factors, but there's still a lot of questions that remain as far as what is the proper exercise prescription? When should exercise be performed? Does it make a difference if it's concomitant with therapy, if it's rehabilitative? And so a lot of work has really tried to look at these effects. And this is just an analysis looking at papers that have used low to moderate intensity, vigorous intensity, compared continuous training versus interval training to really see if, just based on the papers that exist, is there a best therapy that can be your exercise prescription that patients should be performing. And so based on this, they really showed that it's actually more the vigorous training, whether it's continuous or interval, it's vigorous training that kind of needs to be done in order to protect patients against doxorubicin toxicity. The issue with the vigorous training ends up becoming it's a lot more difficult for patients to perform. And so ideally, a lot of different types of HIIT training have been are being developed in order to help to reduce the amount of exercise time and hopefully increase the amount of retention to exercise programs. But based on this, we really wanted to develop a new animal model as well as look at these different training paradigms to see if we're able to recapitulate the patient condition, and then again use this as a model to look at exercise prescriptions and do a lot more mechanistic work that's not able to be done in patients currently. So another excellent graduate student in my lab, Brandon Wynn, has spent a long time really trying to develop what is the best way to model this. Because with the animal, obviously, we have different lifespans. We have, an animal that's going to run on 4 legs instead of run on a treadmill on 2. We have differences in heart rate. We have all kinds of differences between humans and our animals. And so we really tried to minimize the number of things that we couldn't control and model between the 2 species. And so we ended up coming up with was that we thought it was really important to mimic the dosing between the animal and the patient. So we came up with what ends up being in an animal 5.7 milligrams per kilogram, which when you do the dose conversion from animal to human is about 40 milligrams per meter squared, which is a typical dose for breast cancer patients receiving doxorubicin chemotherapy. We went with a 4 treatment schedule, and we have 3 weeks between treatment, again, to try and more mimic what a typical breast cancer patient may receive. And so this also allows us to then take the exercise prescriptions and do them in a way that is identical to, again, what a patient will be performing. So they're able to run 3 days per week, and we're able to also avoid the day after doxorubicin so that we hopefully reduce any type of exacerbation of oxidative damage from the doxorubicin in these animals. And so what he did first was we had this experiment where he had just our sedentary control animals. They did do a little bit of exercise because we looked at their changes in exercise tolerance over time. We have our sedentary dox animals that during this whole treatment period remain sedentary. Our moderate protocol was 3 days per week, 30 minutes per minute, 0% grade, and Then our HIT protocol was also 3 days per week, and it was a 4x4 bout at 45 meters per minute with 3 minutes of active recovery between bouts. And so when we look at these animals, what we saw was that we had a really great survival rate with them. And they all did continue to gain weight over time. So these were young animals. We started this about 10 weeks of age. And so you would expect them to continue to be growing, and they all did kind of plateau towards the end of the experiment. If we look at our heart to tibia length and soleus and plantaris as well, we do see less severity of this protocol at that endpoint, whereas with our bolus dose, we tend to see that reduction in heart to tibia. However, in this case, we were able to maintain that in all of our groups. If we look at the exercise time to fatigue in these animals, we did see kind of this trend for the dox animals to have a reduced time to fatigue and for an increase in the exercise trained animals. And if we actually look at their change in exercise time from beginning of exercise to the end point, we do actually have a large increase in the time to fatigue in the moderate trained dox animals. We then looked at body composition in these animals. We were able to do an echo MRI to get their relative fat mass versus their relative lean mass. And very similar to what's seen in patient populations, you have an increase in fat mass in the dox animals, and that you actually have a reduction in relative fat mass in both the moderate and the HIT animals. And if we look at relative lean mass, we have an increase only within the HIT animals. And so hopefully this helps you kind of think back to earlier in the talk where one of the mechanisms of toxicity was the body composition of the patients, where doxorubicin is going to tend to accumulate in lean mass compared to fat mass. And so this is actually an improved body composition for a patient if you can have increased lean mass, reduced fat mass, and really it's going to help the doxorubicin to not accumulate as much per meter squared. Looking at some of the ventilatory measurements, we didn't actually see any changes in their ventilation at normoxia when we gave them a chemical or a max chemo challenge of a combination of hypercapnia and hypoxia. When we looked at cardiac function, though, we did see a pretty exciting finding, and that was that if we look at their fractional shortening, we did see cardiac dysfunction in our sedentary animals, and that we actually saw that in our moderate animals as well. So even though they are running more and they are having an improvement in body composition, this didn't reflect in their cardiac function. However, in our HIT animals, we did not have a significant reduction in cardiac function. So it appears that at this point, the HIT training is actually the more beneficial training for these animals. Again, similar finding with the posterior wall shortening velocity, we still have that reduction with our moderate doxorubicin treated animals. So the HIT training is by far superior in these measurements compared to the moderate training. And then finally, looking at myocardial performance index, a similar finding, we have an increase with both SED dox and MOD dox and no change in our HIT dox animals. And then kind of finally what we did with these guys is we also looked at their muscle force production. And we do see a little bit of a different phenotype compared to our bolus here as well. We don't see as large of a dysfunction in these animals. The top graph actually shows a direct muscle stimulation. So similar protocol to what was shown in the previous graphs in this presentation. And between these animals, we see no difference in their diaphragm muscle force production. And so this made us kind of curious of, do we have maybe any kind of neural dysfunction in these animals? So we did a separate preparation where we actually stimulated the diaphragm through the phrenic nerve rather than directly. And in this case, we did see some exacerbation or some greater dysfunction in the SED dox animals, just looking at kind of their neural input to the diaphragm. So potentially we have a little bit of neural toxicity that's occurring in this model rather than just maybe direct diaphragm dysfunction. And so we were really excited with the initiation of this model, and we were happy to show that we were able to really recapitulate these clinical findings, that in our sedentary animals, we had an increase in fat mass, we had, no change in lean mass or reduction in lean mass, that they had reduced cardiac function, And so we really thought this is, we did a good job in creating a model that really looked like the patient phenotype. And then also similar, when we look at the studies that have done similar types of moderate and HIT training, we saw very similar changes to what they'd seen as well. So we're pretty excited with this model to move forward with it, to see what kinds of other therapies we can potentially test to see how well we can improve muscle function in our animals. And again, hopefully increase translatability of what we're doing. And so the last thing that I will show real quick is that we have started to look at a difference between concomitant exercise and rehabilitation exercise. And so in this case, we took our same protocol, and instead of euthanizing them a week after their last treatment, we went out in equal duration, and that's when we actually started the moderate exercise training in these animals. And so one of the questions that hopefully you're asking right now is, why did you not do HIT? It looks better. Well, we did, but all the animals died. And so that's kind of brought us back to the drawing board there. It seems that once the animals had already established that cardiotoxicity and that muscle toxicity, that they were very sensitive to that high intensity training. And so we've actually started new studies where we modified the training protocol from that 4x4 to a 10x1, and it has definitely made a difference in survivorship. So that data will be forthcoming at some point. But yeah, this was very interesting to us because again, it seemed like such a improvement in the animal's condition, and so we did not expect that at all. But looking at the moderate though, what we ended up seeing in these rehabilitation animals is that in both the sedentary ones and the moderate ones, that we did have this plateau in the animal's weight gain after they completed their therapies. When we look at some of the muscle to tibia length ratios, in this case, we still didn't see anything with the heart, but we do have a reduction in muscle mass in both of the sedentary dox and moderate dox animals. When we look at their exercise time to fatigue, and then we again also didn't really see any large differences, but if you look at the total distance run from beginning to end of treatment, we do have an increased amount of time in the moderate dox animals when you compare between groups. Looking at relative fat mass and lean mass, again, we have our increase in fat in our sedentary saline, increase as well with our sedentary dox, no change in moderate. Reduction in lean mass with our sedentary saline, same thing with our sed dox, and no change in moderate dox. So as a rehabilitation exercise, the moderate training does appear to have some beneficial effects to body composition in these animals. Looking at cardiac function as well, again, we have this continued reduction in fractional shortening that when we look at our rehabilitation exercise, we're actually able to maintain that better than when we did concomitant exercise with moderate training. Again, showing that both with fractional shortening as well as within our posterior wall shortening velocity. And then finally, what was kind of interesting here too is that when we go out to that longer time point, we do seem to have a difference in the way that our muscle dysfunction is developing. In this case, we did actually have a significant reduction in muscle force production of the diaphragm in our sedentary dox animals, and that was partially attenuated with the moderate training. And we saw, again, dysfunction of the phrenic-induced stimulation, but it didn't seem to be exacerbated when we went out an extra 12 weeks after training or after treatment. And so finally, to summarize this last portion here, is that we're pretty excited about the fact that we can see very positive effects even with this moderate training protocol when it's done as a rehabilitation therapy. Because again, that is a hindrance in the ability of a lot of patients to exercise, is the fact that they are feeling so poorly and they have a lot of nausea and other effects from the chemotherapy. So if we're able to have dysfunction and then reverse some of it, that is a really positive effect, and it seems like something that patients are more inclined to be able to participate in. And so it might be the better direction for some of these exercise therapies to go. So with that, though, I will thank my group and our funding sources, and I'll thank you all for your attention, and I'm happy to take any questions.
Sorry if you don't know the answer to this, but looking at those pit mice that died,
Yeah.
was that like during the high intensity interval training that they like had used, it was like an actual like heart attack kind of thing?
So it was a mix. It was a mix, yeah. Some of them were just found in their cage, and other ones did actually have an event while on the treadmill. So yeah. And we ended up switching to, so we started with the 4x4 because our clinical collaborator, that's what she was doing a lot with her older patients who are performing exercise training. And so it's pretty standard protocol. But the 10x1 is actually something that some groups working with chemotherapy patients have started to use, because it is a lot easier for the patients to do, having that just 1 minute bout. And so it showed the same thing within our rats as well, that those shorter but more intervals is kind of more optimal for chemotherapy patients. Yes, no, you're good.
Two questions. I'll ask you a question first. The timing, right? You did before treatment, and then you still prehab versus rehab. Have you tried anything, and that was what I think, to start it with the treatment, or even at the same time as the onset? I mean, I think it's probably like the rehab or the exercise is probably more prevalent than potentially the earth. Why do you think that timing you think, you say the damage muscle, it's just like rather than particular. What's the real timing? How much training do you need before the
So yeah, we
just go and say like if the books treatment not the day after the 2 years. I started exercising 2 years.
So that's definitely something we haven't modeled in our animals, but some of the people clinically working on this have tried to see, are we able to do 1 bout of exercise the day before treatment, and can that provide some beneficial effects? And it appears that it can. There are definitely smaller effects, but there is some positive benefit to even just 1 bout of exercise the day prior to treatment. The most we've done right now is the new project where we started doing the 10x1. We took all of the animals out through the rehabilitation period, and so the concomitant ones actually continued to exercise, and they weren't limited by the fact that they had trained during treatment. We also actually started doing VO2 max tests on the animals too, so that their prescription is actually closer to that 70 and 90% that we estimate for what the training is supposed to be. And we did modify it over time. So these animals did a pre-VO2, they did 1 mid-treatment to see if we needed to increase their exercise intensity. And so we were able to give a much more precise exercise prescription to each group. And so that I think really made a big difference in kind of the way we were able to control and translate this.
Intermittent fasting during the therapy is actually intermittent fasting before it's beneficial, and it's sort of like it's really good because it doesn't really seem depending on what the onset of that is. So
yeah,
right before the exercise, then done with it.
Yeah. I mean, timing and intensity is so important. And again, that's part of the reason why people are really scared to do too much exercise close after the doxorubicin treatment, is because you have such a large increase in reactive oxygen species and inflammation and things after the doxorubicin, that then adding the stress of exercise, they really think that it's actually going to cause a lot more dysfunction rather than kind of those positive adaptations of exercise. So you have to be very, very careful with timing of things.
Very nice stuff, actually. In your rehabilitation studies, I noticed that the moderate intensity exercise group also show a little bit of less survival than the animals that were kept sedentary.
Yes.
Is there any concern about that? Even moderate intensity exercise also increases mortality rates?
So I think there definitely is a concern, and we've seen this with a couple of different models that we've used. We have another collaborator who was rehabilitating mice that had received FOLFOX chemotherapy, and you actually kind of saw this stair step where things were exacerbated in the exercise animals, similar to the FOLFOX treated animals, just because it was probably too high of an intensity to actually have the benefit they were looking for, and especially with the actual chemotherapy insult. So there definitely can be too much. In this case, I'm not 100% sure because when we redid the very similar study, our mortality rate is much better. So a lot of it too is even just the infusion of the doxorubicin, because you can have a lot of toxicity that occurs just from extravasation of the doxorubicin as you're giving the infusion. So I think we've improved our techniques as well as kind of improving the exercise prescription a little bit better by actually having their VO2s, I think is a lot of the reason we see better survival now.
So similar to what she asked, I was just curious, counterintuitive, but I mean, doxorubicin is totally toxic and it's meant to be toxic, it's supposed to kill the bad, save the rest.
Yeah.
Right? So in my mind, if you're, exercise is protective of the skeletal muscle and cardiac cell, is there any thought that like it's going to create some adaptation to cancer cells and somehow make them more resistant to toxicity?
Yeah, and so that's something that's always been difficult with a lot of the work related to any type of chemotherapy is that you need to maintain its anti-tumor efficacy as well as protect the other tissues. And so that's where maybe rehabilitation therapy is actually a better thing if there is some type of adaptation there. We haven't done any of the tumor-based studies, but most of the groups that have and they've combined it with exercise actually see either no difference in tumor size or they see an improvement in tumor reduction. So to my knowledge, there's not any type of adverse effect on tumor growth with exercise. But yeah, obviously it's something that is a concern, and any type of intervention really needs to always consider the anti-tumor activity of the drugs.
I was going to ask a similar thing. If you're increasing energy expenditure, everything's turning over faster with
Yeah.
exercise training. Are the kinetics, the pharmacokinetics, they must be working if there's no detriment to
Yeah.
exercise.
Yeah, and so there's also a little bit of work that's being done that has looked a little bit at the tumor itself and thinks that there's maybe kind of increased blood flow to the tumor, so that potentially that's why you're seeing increased delivery of the doxorubicin. So you might actually be allowing it to localize to where it should be a little bit better. But yeah, a lot of
You're also reminding me, I think of what's good for people, and there are good diets and there's good physical activity, and we need to be really careful to not think physical activity is good all the time. Who are you talking to? Are they a cancer patient? Do you know what I mean?
yeah.
We somehow think our good health prescriptions are universal, and this is a perfect example where they might not be.
Yeah, and I think that's definitely something that kind of has slowed the field a little bit, is the fact that the patients are also different. And
Yeah.
so the prescription also needs to be very different based on that. So it's tough.
So if the mass is protected, protocols, versus cardiovascular disease.
Yeah, so the trouble with animals and resistance training is always that there's not great models for that. And so we would love to look at some of those things where you're able to improve kind of muscle mass, but we've not settled on a exercise that we think could do that yet. I think a lot of it, and it just complicates things in the clinic, is really just making sure that you're basing doses on lean mass rather than total body surface area. And that's something where, people just seem to be getting overdosed because they think in general more is better, but obviously in this case more is more toxic. So.
The muscle function is that put the maximum strength.
Mhm.
This may not be the best outcome, because we, for instance, we did just recently finish a study on cardiorespiratory study. We find the same maximum strength, but we do 50 repeats of maximum voluntary contraction. Cardiorespiratory actually lose the strength, so the decline in strength with the repeat exercise, or exercise with the repeat contractions is faster than cardiorespiratory. And so that's what we get solutions, right? Because people really need one maximum contraction.
Right.
Fatigue for the fatigue. That's probably potentially one of the mechanisms.
Yeah, and so we didn't in this study because when you do the phrenic nerve preparation with the direct muscle stimulation, you basically are end up doing a fatiguing protocol because we looked at neuromuscular transmission failure. But in the second study that we did, we took away the nerve question, and so we have the fatiguing protocol in those animals. I haven't seen the data yet, but I'm excited too. And we did start to do that a lot more with our acute animals, is really to not just look at what is your absolute strength at these frequencies, but are you having increased fatigue compared to the exercise or the sedentary animals? So.
I think that's it.